Thanks, Helga. So I'm going to just give you an overview, really, in the next 20 minutes of where we are in the field of TB vaccine development, with a particular focus on what I see as some of the challenges for us. So TB needs very little introduction these days, particularly to this kind of audience. Although when I started working on TB 15 years ago, many people came up to me and said, haven't we eradicated TB yet? And I think what's interesting is no one says that anymore. And I think there is much greater global awareness that we have never got close to eradicating TB, and I don't think are going to for generations to come. So the latest figures are up here, 9 million new cases in 2013 and 1.5 million deaths. And clearly the emergence of drug resistance, particularly multi-drug resistant, extensively drug resistant, and now totally drug resistant strains of TB have compounded the problem and made our ability to treat this disease harder than ever. Geographically, the overlap with the HIV epidemic has had a devastating impact, particularly in sub-Saharan Africa. And as if that all weren't enough, the burden of latent infection is that about a third of the world's population are latently infected with MTB, and clearly are at risk of reactivation should that latent infection, should they become immunocompromised for any reason. And globally, of course, the commonest cause of immunosuppression is co-infection with HIV. So TB is a bit different to malaria and HIV, other pathogens of enormous global importance, in that we already have a vaccine, BCG, which is one of the world's most widely used vaccines. It's been given to over 3 billion people throughout the world. And BCG is live attenuated Mycobacterium bovis, the strain of Mycobacterium that preferentially infects cattle, and the reason why we pasteurize our milk. BCG has been around for almost 100 years, and there have been many, many efficacy trials looking at how good BCG is at preventing TB. And when you put all those trials together, there are some very consistent messages. BCG, when given at birth, as it is throughout most of the world, does confer consistent and reliable protection against TB meningitis, disseminated TB that is spread outside of the body. It also seems to protect against leprosy caused by Mycobacterium leprae. But what it really doesn't do, particularly in high-burden countries, is protect against lung disease. And that's really where the burden of disease is and why we need a better vaccine. There was a very interesting study published by Lara Rodriguez and her colleagues from the London School several years ago now, where they showed that boosting with BCG doesn't make things any better. So Lara took 200,000 Brazilian school children, all of whom had had BCG at birth, and she randomized those children by unit of school to either be revaccinated with BCG in adolescence or not, and saw no impact of revaccination on reducing incidence of TB. So we need to do something better than just give BCG many times. So pulling all this together, how do we go about designing a new TB vaccine? Well, sort of briefly and in overview, because of that protection that BCG confers against disseminated disease in the first 10 years of life, really all the current development strategies include BCG or another live mycobacterial priming agent in some shape or form in those strategies. The current thought is that we need to induce a potent Th1 cell-mediated immune response, because although we don't have a correlate of protection, and to have a correlate is very much a holy grail in this field, and would transform the landscape in terms of vaccine development. We do know there's a lot of evidence that the different components of a Th1 cell-mediated immune response, interferon gamma, CD4 T cells, TNF, are essential in protective immunity against TB. There is a resurgence of interest at the moment in the field in the induction of antibodies. So the field goes in cycles in this, from saying that antibodies have absolutely no role to play to, well, antibodies may have some role to play. And we're currently at a point in that cycle where we think antibodies may have some role to play, particularly if you're thinking about prevention of infection rather than prevention of disease. And I'll come back to that when I talk about efficacy trials. So putting all this together, there are really two main strategies that are being focused on. Either you leave BCG as it is, given at birth throughout the developing world, and develop a subunit booster vaccine, be it a viral vector or a protein adjuvant combination, to be given at a later point after BCG, either later on in infancy or perhaps in adolescence when we know the effects of BCG are starting to wane. An alternative strategy is that you take BCG itself and make it better by genetically engineering BCG to preferentially access different immune processing pathways, or perhaps there is a school of thought that BCG is the wrong starting organism and we should be rationally attenuating Mycobacterium tuberculosis. And the first prototype of that class of vaccine has now been through a phase 1 study. And clearly you can put the two together, so you could boost or enhance an improved BCG. So from the theory to the reality, this slide shows all the different candidate vaccines that are currently being tested in the clinic. And the details are not important, but I think there are a few messages from this slide. Firstly, if I'd put this slide up 12 years ago, it would have been a blank slide. So 12 years ago, there were no new TB vaccines being tested in the clinic, compared with about 50 for malaria and about 50 for HIV. So that gives you a sense of the consequences of the decades of neglect in TB research. The other comment about this slide is that there are different color boxes which represent different classes of vaccines. So you have whole mycobacterial vaccines, such as MVACI, such as VPM1002, which is a recombinant BCG. You have viral vectors, such as MVA, the vaccine we developed in Oxford, and the Crucell Ad35. And then you have protein adjuvant vaccines, such as M72, Hybrid 1, Hybrid 56, and Hybrid 4. So there are many different classes of vaccines, and people are testing at the moment to really try and induce potent, particularly cell-mediated immunity. But as I say, there is also a focus on inducing humoral immunity as well. So just a little bit on one of the vaccines we developed in my lab in Oxford, really just to illustrate the pathway for development and some of the challenges. So MVA-85A is an example of a subunit booster vaccine. This is a recombinant viral vector, so the virus bit is MVA, which is an attenuated strain of vaccinia, the smallpox vaccine, which expresses antigen 85A, so a very immunodominant antigen conserved across all mycobacterial species. And the aim of this was to boost the effects of BCG. After we'd done a lot of preclinical animal studies, we then moved into the clinical program, and this was the first candidate TB vaccine to move into clinical testing anywhere in the world. The outcome measures in those clinical trials were obviously safety, particularly in the first studies. We've done an enormous amount of work looking at immunogenicity, so what kind of immune response are we inducing with this vaccine? But ultimately, the bottom line is efficacy. Does this vaccine stop people getting TB? So 2 years ago now, we published the results of the first efficacy trial. So this was an infant efficacy trial, and it was an infant efficacy trial because the disease burden, TB disease burden in infants is enormous, particularly in Africa. So we took 3,000 babies or thereabouts who were all vaccinated with BCG, and then they were randomized at 4 to 6 months of age to either be boosted with MVA or to receive placebo vaccine. We followed those infants up for every 3 months until the study completion. And it's essential in this kind of study to capture all your endpoints because you're always at the edges of power on this kind of study. So we had a setup where every single baby that had any TB exposure or any symptoms was admitted to a dedicated case verification ward for investigation. And over the course of the study, we had over 1,000 admissions in this ward. So you can see and imagine what an enormous workload that generated. At one point, we actually had a waiting list of infants to go into the ward. I mean, they weren't sick, but it gave you a sense of the volume. The trial was powered on disease to see a 60% improvement over BCG alone. Now that's a big bar. WHO modeling figures show that if you had a 40% improvement in BCG, it would have an impact globally on the epidemic, particularly if you used a catch-up campaign. But you couldn't do the study. The study would just be too big. And we also had MTB infection as an endpoint using quantiferon conversion as a diagnosis of infection. So to cut to the chase, the endpoints are here. So our primary efficacy endpoint against disease, this was our most stringent endpoint. We saw 39 cases in the placebo arm and 32 cases in the vaccine arm. So this gave us a non-significant vaccine efficacy of 17% with a confidence interval that spans 0. We saw the same story with the other endpoints, and particularly with the quantiferon conversion, where we had over 350 endpoints. So we didn't see efficacy, which was obviously an enormously disappointing result for us and for the field. But it's absolutely critical that we try and understand why we didn't see efficacy and what we can learn from this trial to refine both the vaccines that we go on to develop and the way we do trials and the way we select vaccines. So one of the possible reasons why we didn't see efficacy in this study was that the immunogenicity was actually pretty disappointing. So if you give the dose of vaccine that we gave in this study to UK adults, you see an median immune response of about 1,650 spots per million. This is a way of measuring antigen-specific T cells. That compares with the median response in this infant study of 136. So you can see there's an enormous magnitude of a log reduction in immunogenicity. Now, we don't know if that's why we didn't see efficacy, but I think it is a relevant thought. We don't know if more of this, if we'd seen this level of immunogenicity in this infant study, would we have seen efficacy, or do we need something that is qualitatively different? That's not a question we can answer with the data we have at the moment. So what can we learn from this trial? Well, clearly one of the most important questions is, why is the immunogenicity reduced? This is not the only vaccine where we see a reduction in immunogenicity as you move from developed world adults through to developing world infants. And there are clearly many factors that may be contributing to that. But we need to try and understand more about that and develop vaccines that overcome those issues. And what about the correlates? Well, I'll talk about those in a second. I think one of the important things is, how can we make the preclinical animal models more predictive? So there are many different preclinical animal models that are used for TB vaccine development, and we need to refine our view of those models and try and make them more predictive. And some of the ways that are being considered are using natural transmission models rather than giving single large challenge doses, which of course doesn't really represent what happens in humans. Using clinical isolates to test those vaccines in animals rather than using highly passaged laboratory strains, and raising the bar. Actually, we probably need to design vaccines that are much better in those animals than we're currently doing. We clearly need to make some better vaccines. And we also need to improve the criteria by which we select which vaccines progress. Whilst it's fantastic to have that pipeline I've showed you of vaccines coming through, we cannot possibly test all those vaccines in efficacy trials. So we have to have a rational, objective selection process by which we select which vaccines should go to these very large, very expensive efficacy trials. One of the things that's come out of our infant study is a refocus on the best, or not the best, but perhaps the most important and most tangible efficacy trial that should be done, and the most tangible target population. We did that efficacy trial in infants because at the time it was the only efficacy trial possible to do, and infants are an important target population. They have a very big burden of disease. But actually, adolescents and adults, whilst on average they have about 50% of the incidence of infants in the developing world, they are responsible for most of the transmission. So infants don't develop smear-positive disease, so they don't transmit, whereas adolescents and adults do. So potentially you can have a much bigger impact on the epidemic if you protect your transmitting population. And of course, one of the reasons for this shift is that many of the vaccine candidates are looking less immunogenic in infants. The other shift has been towards a focus on prevention of infection rather than prevention of disease. So the obvious reason for this is that it's a faster and therefore cheaper trial, as there are many more endpoints. So you'll notice in our infant trial we had 71 disease endpoints, but we had 350 infection endpoints. So you've got much greater power to see an effect. Now that's great. But I think an enormous caveat that we simply don't know the answer to is, are the protection mechanisms the same? Will a vaccine that protects against infection necessarily protect against disease? And importantly, is the converse true? Because what we don't want to do is throw out a vaccine that doesn't work against infection, ignoring the fact that it might or might not work against disease. There's also a caveat here about how we define infection. Correlates of risk are really important. So in our infant study, we collected samples from every baby that took part in that trial with the aim to look for correlates of protection. We didn't see correlates of protection, but we do still have cases and controls. So we can look for what's called correlates of risk and look for immune responses in the infants that did not go on to develop disease compared with the immune responses in the infants that did go on to develop disease, with the idea that that might guide vaccine design. So we've just finished an enormous amount amount of laboratory work in my group at the moment looking at this. So we had 284 infants, 2 samples per infant, and we run a whole series of assays on a single vial of cells. So you can see this had to be incredibly carefully choreographed because you don't get much blood out of babies. We've done transcriptional analysis, functional analysis with growth inhibition assays, conventional ELISpots and antibodies, and we've also looked at cell surface phenotyping. So that data is being analysed at the moment and will soon be submitted for publication. Okay, so briefly just to finish, one of the things I said at the start is we need to make some better vaccines. And briefly, there are 3 ways we're looking at this in my group. So the first is to look at new routes of immunisation. The second is prime boost combinations, putting together different viral vectors that may induce synergistic immunity. And the third is looking at new antigens. It may be over-optimistic to think that one of the 4,000 or so antigens that mycobacteria have will be sufficient to protect outbred humans. And just to focus on one of those, an inhaled TB vaccine. So it's obvious when you think about it, TB enters the body through the lungs, and it may well be that the best way to protect against TB is to design a vaccine that is given into the lungs and induces local mucosal immunity. There's some animal data to suggest this, and of course inhalation is a very common route of drug delivery for asthma and COPD. It's important to say this is not a new idea. There's a wonderful paper from 1968 in the literature where Rosenthal et al. Locked a group of medical students in a room, I'm not sure it was with fully informed consent, sellotaped the windows and the doors, I think locked the doors, and nebulized BCG, and then looked at skin test conversion. And actually most of those students did skin test convert. So we did a study which was to GCP and was with fully informed consent, where we took 22 BCG vaccinated adults and we randomized them to receive low-dose MVA. This is the first time anyone's done this in the world, either by aerosol or intradermally. All subjects then had a bronchoscopy, so we could take mucosal washings from the lungs and look at mucosal immunity, as well as take a blood test and look at responses in the blood. Obviously, safety was an important outcome here. I'm not going to show you the safety data because it was fine, but I am going to show you one slide on the immunology. So here we've looked at the bowel, the bronchoalveolar lavage fluid, and we've looked in the blood, and we've looked at antigen-specific T cells. And what we see is when you compare aerosol administration with intradermal administration, looking at 4Th1 cytokines, you see higher levels of lung immunity when you've given the vaccine by aerosol compared with in the arm. Well, that sort of makes sense. Although, interestingly, it's not 0 when you give the vaccine in the arm. But what's really important is when you look in the blood, the immune responses after aerosol are at least as high. So we're not in any sense compromising the systemic immunity by giving the vaccine into the lungs. And I think this is because this is a very potent route of drug delivery. You have a surface area the size of your tennis court in your lungs for drug absorption. So we're doing some more studies on this. And then finally, and this is my last point, I think one of the other things we need in the field are better models. So I have the luxury of working in Oxford with a very big malaria vaccine group, and they test their vaccines by challenging people with malaria. So Adrian Hill will vaccinate 50 people with his malaria vaccine, and then he'll take them off to London to Imperial College and give them malaria. They're bitten by 5 infected mosquitoes. And that's a way to test if these vaccines work before you move to these enormous field efficacy studies. Now, we can't give people TB for pretty obvious reasons, but we can give people BCG. And BCG is a live replicating mycobacteria. So can we somehow use that as a model to test our vaccines before we, just as something to add to the animal models and the immunology, before we move to these field studies? So we're doing a trial, or in fact we're now on our 3rd trial, where we're vaccinating people or not, and then we're giving them BCG, and then we're taking a biopsy 2 weeks later from that BCG site, and then we're quantifying the amount of BCG in that biopsy site to see, and the idea is that the group that have had the vaccine will have less BCG in that biopsy than the group that haven't had the vaccine. And here you can see looking at culture, so group A is a naive group and group D have had BCG followed by MVA, and you can see a significant reduction in culturable bacilli using that model, and you can see that the 2 groups that are naive to BCG compared with the 2 groups that have had BCG have a significant reduction when we measure this by PCR. So there may be a potential here for a model that could add to our algorithm for vaccine selection. So I think that is very much a whistle-stop tour through where we are in the field and where we are in my group. I think in the last 15 years, 20 years, there has been an enormous amount of progress in this field in terms of vaccine development. We now have over a dozen candidates being tested in the clinic. We really urgently need better models, both animal models and human models. Humans are the target species here. We must use every opportunity to identify correlates because that would transform the landscape and make this all so much easier. We must use the efficacy data to review and refine these models in an iterative way, because that's the only way we'll make progress in this field. We'd better make some better vaccines, and I'm afraid my bottom line is there is currently no substitute for human efficacy testing. Just to thank the enormous number of people whose work I have whistled through in the last 20 minutes. So my group in Oxford, both past and present, my collaboration with the respiratory unit who do the bronchoscopies, Helen Fletcher, a very close colleague and now collaborator at the London School, and all the many partners and funders, including the Wellcome Trust, including NORAD and TBVI, who fund, and ERAS, who fund and contribute to this work. Thank you. No?
Thank you very much. I really appreciated your talk. Just a silly question.
No, it won't be.
When you do bronchoalveolar lavage, there actually for chronic obstructive lung disease has been shown gender differences in the types of cells responding. So when it comes to TB, are there any gender or ethnic differences in cellular immune response?
Yeah, that's a really good question. There is a small literature on gender differences to vaccine responses as well, actually, not TB vaccines per se, but across the board of vaccines. So our subjects in this study are male and female, and when we've looked, we see no differences. I mean, the numbers are relatively small, but it's a good question, and it's something we should. Ethnic, okay, so most of our Oxford volunteers are Caucasian, white. Some of the volunteers, obviously our African studies, it's more mixed. It's a good question, and I think it'll be interesting to do more comparative studies looking at these factors. Yeah.
Thank you for an excellent lecture. I wondered about, you said you'd come back to latency and some evidence for BCG in prevention of infection. I thought was interesting in terms of the household contact studies.
Yeah, so I mean, the dogma has always been in the field, as you know, that BCG does not protect against infection. And of course, the literature that it protects against disease is very complicated. There have been an increasing number of studies, largely from not high burden countries, I think, largely from low and middle burden countries, suggesting that BCG may protect against infection, which I think has led to an acceleration in thinking that actually that is a more tractable target because the power is greater and therefore we can do smaller studies. I think I am concerned whether the mechanisms are the same. My own bias is you probably need antibodies to protect against infection, and so you may be looking at a different immunity, and certainly I think the HIV field would suggest that is the case. I think the biggest caveat is the tools we have to define infection. You know, we define infection using an IGRA, quantiferon gold or T-spot, as an immune response to TB relatively specific antigens. You know, that's a big step away from the pathogen. And I think we don't fully understand what happens if you eradicate your IGRA response. Is it just below the limit of detection, or have you cleared it? Have you eradicated that infection? We just don't know. So I think we're limited by the tools we have to define that endpoint.